Great. Thank you, Julie. And thank you all for being here today. It's a pleasure to introduce Lottie Jensen, who is giving a talk today. Lottie, I am just at Western Washington University for a year, and so I wasn't necessarily planning on advising any honors students. But I was approached by Lottie this fall after Dr. Catherine Sobosinski recommended she get in touch with me to advise her on this project. My background is wildlife biology, and I've done a lot of telemetry work, mostly with birds, but the patterns are a little bit the same for mammals or birds or anything you're tracking. And so after meeting Lottie, I was pretty excited about the project, and she seemed really dedicated and hardworking. And it's just been a pleasure working with her. She's been very self-motivated and had a really great attitude towards this project, not trying to take on too much, but also really doing some solid work and learning along the way. So she is going to tell you all about her links and what they're up to as sort of learned through this telemetry work. So Lottie, take it away.
Thank you very much. Thank you guys for coming to my presentation. This is kind of a labor of love of like lots of different people smushed together, which I personally think is very exciting. And I think adds a lot of depth to this. So I just want to give a huge shout out to Home Range Wildlife Research, because this is their data that I'm using. And as I'll get into it a little bit more, I was able to work with them for a summer. So that's how I sort of got ingrained into this work. But yeah, I just wanted to give a huge shout out and thank you to them off the bat. Okay, so diving in a little bit to the Canada lynx. It's a very charismatic animal, very cute. I had it on the main slide there. It's a medium-sized carnivore, a cat. Lynx canadensis is the scientific name. There are native populations of lynx throughout the northern United States, Canada, Alaska. They are severely declined in their population in their southern reaches. So that would be down here in the contiguous US and then also in southern Alaska. In fact, in 2000, the US listed them as threatened under the Endangered Species Act. And in 2016, Washington listed them as endangered. So the Washington population, you can see a little bit up here in Unit 4. That is the unit and the population that my research and Home Range's research is based on. That's kind of it's in Okanagan County, right to the east of the North Cascades. If any of you have ever been to like Winthrop, the like Western cowboy themed town, it's right in there. And in the state of Washington, so within Unit 4, which is designated as critical habitat under the Endangered Species Act, there are also lynx elsewhere, and there's estimated to be about 50 to 100 right now in the state of Washington. Lynx are pretty specialized cats. They like their habitat that they like, and they don't really want to move outside of it. So they really prefer high altitude, boreal forest. So that's stuff with more evergreen trees, a fair amount of disturbance moving through it, and super reliable snowpack. They really like the snow. This is a trail camera picture from Home Range. There are 2 lynx in this picture. There are 2 kittens, actually. The mama has just moved away in this image, but they like the snow. It keeps them really quiet, helps them sneak up on their prey of choice, which would be snowshoe hare, which is their main diet. And so what's kind of interesting about Canada lynx is that we can see that their population follows really closely with snowshoe hare population. And if any of you have taken kind of like Ecology 101, you probably did population maps that had lynx and snowshoe hare following each other, so they really rely on strong populations of those hares to live. I'm going to talk a little bit about Home Range Wildlife Research, as well as my job there a couple summers ago, which was a wildlife field technician, just because this has really informed kind of what I was interested in doing in college moving forward, and hopefully what I can do in my career. It was a really amazing opportunity. I got through the Sustainability Pathways Fellowship at Western, where I was able to intern with Home Range for a little bit. This is Carmen and Anna. They were my lovely bosses, and they're headlining the Lynx and Wildfire study at Home Range right now. And that study is diving into the tripod complex fire, which was a fire that occurred in the Pasayten Wilderness in that Unit 4 of Critical Habitat in 2006. And at the time, that fire was one of the largest in Washington's history. And this is kind of mapping it out a little bit. Elevation, and then just all the little points where there are fire starts along the way. This was a map I got from Home Range and some of their talks that they've done. And so within this research, they're capturing and collaring Canada lynx. Here's one in a box trap. Very cute little guy. So they're capturing those lynx and then they're tracking their movements. And my work was to go back during the summer over areas that they had tracked lynx the winter before to do vegetation plots. So I was participating more in the habitat assessment side of things, where I was looking at how many trees there are in a set area, what's the size of the trees, what's the primary vegetation undergrowth. And these are just some statistics, just little deliverables I got from working there that summer. I was working with a couple other interns as well as Carmen and Anna. We did almost 600 vegetation plots. Those included tracks we went back over where lynx were that winter and random ones. Counted a lot of trees, tagged a lot of wildlife photos. And as I'll mention a little bit later, we found a lynx den, which was really exciting, and we got to go see it. Yeah, so these are also just some beautiful views that I got working out in that area over the summer. It's really amazing. Those are some wolf tracks. So even when you're on the roads, there's big wildlife walking around. These are just extra fun little things from my summer. This is me at the lynx den with one of my other interns. This is me tagging a rattlesnake, which isn't related to my project, but I personally think it was one of the cooler things that I did. And I like to say it as kind of a party trick, that I can tag rattlesnakes. It's pretty cool. And then this is just the coolest wildlife trail camera picture I think I've ever seen. It's a little gruesome. But yeah, it's just a wolf with a baby fawn. And it's just cool to you're out there, doing research in the backcountry, but you don't really see a lot of animals when you're doing that because you have heavy packs and boots and you're tromping around and making a ton of noise. So it was really cool to go back through the trail cam pictures and like be able to be like, oh, I've been there and these animals are also there. So that kind of makes the work feel a little bit more meaningful, even if you're not seeing things when, you're out there doing the work yourself. Okay, so to get into what I'm doing for this particular project, like I mentioned, at Home Range, they're capturing lynx and collaring them with GPS collars. Those collars are transmitting to satellites and giving us GPS location data that's called telemetry data. And in 2025, they were able to collar 5 lynx. So it was 3 males and 2 females. I will be using their names in this presentation. So Freddie, Oscar, Buster, Honey, and Pippi. And this is just a little bit of statistics about things from their capture. So a little bit of demographic data, 3 males, 2 females. They're all about the same size. Honey is the smallest, also one of the youngest. Oscar is huge and one of the youngest, and that will kind of come into play later when we look at his movements. And Pippi is older. Her location is also much further south than the other lynx. But yeah, so the GPS was transmitting every 30 minutes. So I have up-to-date locations for every 30 minutes for these links for the month of March. So that's awesome. And that will kind of be what we'll look at going forwards. Okay, I also want to give a little introduction into GIS and ArcGIS for those of you who might not be familiar with it. This is a software for making and designing maps. This is the user interface for ArcGIS Pro, which I can get through the school, which is super duper awesome. Mainly for my project, I was using this side, which is geoprocessing, and these are just a bunch of tools that are in the GIS software itself. And I can go in, click which tool I want to use, input a bunch of information about my data and how I want it to look, and it spits out like a beautiful image for me, or a beautiful analysis. And from there, I can make it look the way I want to look with more cartographical skills, which I wasn't doing in this project. This is mostly analysis, but you can make graphs look really, or maps look really pretty in GIS if you want to. And as I move into my methods, I'll point this out over here. This is kind of just like what the data looks like when you put it into GIS. So when I first put information in, when I first get a data sheet, I put it into GIS, it's gonna look like this. And then this sort of lump is what I'm gonna use when I do my analysis going forwards. We'll talk a little bit about what I did. So for my data, I received a shapefile, which is what GIS just likes to process. And it included things like the Lynx ID, which is the names you guys saw earlier, the caller ID number, GPS date and time, so that's when are we receiving these points, and then the latitude and longitude of the Lynx location. And so that kind of gets into my main goal for this project was really just to be able to practice moving telemetry data, which is just a latitude and a longitude, and making it something that is easy for me to understand and for other people to understand, something that's more ecologically meaningful, where you can really look at it and be like, oh, this is what these animals are doing, and maybe come to some conclusions based off of their movement. Because just looking at a latitude and a longitude doesn't make a ton of sense. So my goal was to create one map with unedited collection data, so that would be all the links together. And then I wanted to separate the links individually to look at their movements, like as individual links, and see what was going on. So then ArcGIS Pro, I used the space-time kernel density tool for this analysis. And so that just sort of allows me to look at the density of location points over a given time. And so I'll show you a little bit what that looks like, because I think it's easier to understand when you're really looking at it. But this is just explaining what I put into the fields in the geoprocessing pane. And the important thing just to mention on here is that within space-time kernel density, you could do an elevation field or a time field. I chose to use time because I have those locations as like 3:30 p.m. on March 14th. So by using the time field, it allows us to get that into the final analysis. And then, so I used that for the unedited collection data, and then I also used that after I separated the links into individual data sets. So I did one space-time kernel density analysis on each individual links and just put that into one map, which I'll show you guys. But this is the output from unedited collection data. So this is all of the links together. And I'll explain a little bit about what this space-time kernel density really means. So I think this is a really good example of a time when the links aren't moving around as much. And we can tell they're not moving around as much because of the bluer color. That means they're just a little bit more stagnant. The redder the color is, the more dispersed the location points are, the less densely clustered together. And so this would be an example of a time when they're moving around a lot. So these are the boundaries of where they're moving. And then the red just means that like they're moving around quickly within the boundaries. And then over here it would be like, these are the boundaries where they have moved within this time, but they're more likely to be staying in a spot in the middle. And so this is just an example of some different times. This is midnight. They're not moving a ton. They might be sleeping. They might just be hanging out. This is the middle of the day, moving around a lot, probably hunting, probably, tracking the extent of their home ranges. But for me, it was a little bit hard to piece this out without like looking at the individual links. But I also wanted to show you, this is a little video of what the tool ends up looking like. So this is with the time component. So this is the sliding bar is over the course of the whole month, and you can just see how their movements change a little bit. The one thing to point out is that, let's see if I can just like get to, yeah. Okay, the one thing to point out is that this is Pippi, and this is only Pippi, and you will see that later. This is all the other 4 links, and this is just Pippi. So nobody else is coming down here, and Pippi's not going up there. She likes her little spot down at the very southern reaches. But yeah, you can just see it over the course of the month. I think it's kind of interesting just to look at how things change. And Pippi's data was collected, I think, March 13th through the 22nd. So it wasn't for the whole month. That's why she shows up at the bottom halfway through and then goes away again. But it still gives you a pretty good idea of like how things are changing, when they're moving, when they're not. Yeah. Okay. And here is them individually. So I'm gonna take us to a point where we can see Pippi too. Okay. I tried to color code it a little bit, but I will piece it out. This is Freddie. This is Buster. This is Oscar. He is our young and our big one. And you can see that when you're looking that he is moving a lot farther and a lot faster than the other ones. This is Honey, and then this is Pippi down here. So it's the same sort of deal. The darker the color, the less they're moving. The lighter the color, the more they're moving. And I'll just play this video out from the top so you can see how it all changes over the month with those individual links. You can see Oscar's kind of going all over the place. And I'll explain a little bit more also about my observations with this data and what I really took away from looking at these movements. Yeah, and then Pippi goes away. All right. Another thing I wanted to add is I was able to get capture data from Carmen at home range. And so I wanted to add the capture sites onto the map of their locations, just because I think it adds an extra level of, just because we see that these links are moving this way over the course of the month, it doesn't mean that they're not going other places too. So we can kind of see this. This is just a snapshot in the time month span of the link's movements. That star up there on the left is Buster's capture location. That one on the right is Oscar's. This is Pippi's, obviously. And then Freddie and Honey were captured in the same place with the same trap. So that's why there's only 4 stars when there are 5 links. But I just kind of wanted to exemplify that because in this we can see that, in this range of time, Oscar and Pippi are moving within their capture locations, but the other 3 aren't really. Especially Freddie and Honey were captured further south and further for Honey, further west than she really moves. So that's just like, this is a snapshot of time. This isn't exactly where they're moving all the time. It's not going to be perfect. Obviously, we don't have cameras trained on them to know exactly where they're going all the time. This is the most exciting part, and this gave rise to the title of my presentation, Lynx in Love. One of the most exciting conclusions I came to when looking at my data is that Honey and Oscar were going in the same place at the same time, which is unusual because lynx are solitary cats. They like their home ranges and they don't often overlap. And these cats are the same age, and it is March, which is breeding season for lynx. So Audrey and I kind of came to the conclusion that it made sense that they're probably a mating pair. So I'll show this little video of just the time in which it's overlapping. It's over the course of a couple weeks. Lynx, male lynx specifically, also will follow female lynx around for a little bit before mating, and then they'll mate a few times over the course of a week and then often go their separate ways, but come back together for kittens as well. So I'll just show you this video because I think it's very sweet. You can see they're kind of solitary and then they move together and then they're going to move apart for a little bit and then come back together again towards the end of the month and kind of just hang out around each other and in the same place, which is just kind of cute. I think it's kind of cute. Maybe we'll get some good kittens out of it, hopefully. Okay, so in terms of my observations, I wanted to start with the individual lynx first and then move on to group dynamics. So big thing is that Oscar is moving very far, very fast. He's the youngest and the biggest, thereby his energetic needs in terms of food are probably more than the other cats. He's still growing and he's growing big. So he might need more food and he might also not be as good of a hunter as some of the older lynx. So he might need to go further to find what he needs. Another interesting thing I noticed is that Freddie and Buster, our two male cats towards the west of the data, have home ranges that kind of border each other. And the interesting thing about that is that that border, when you zoom in really close, is a creek. It's 20-mile creek. So they probably are just hanging out and around the same place. They both don't move very far and very much, so it's likely that it's really productive for them. They're able to get all of the food they need in that location, but they don't really want to cross the creek to get into each other's zones. Yeah, and like I said, Buster doesn't move much in the second half of the month. This could also be for many reasons. He might have just found a large food source and he doesn't need to move from it. Like I said, lynx primarily like to eat hare, but they will eat other things if hare are not available to them, like small deer if they can get them. And they can stash their food and live off of it for a little bit. So it's possible that that's what Buster is doing. And Pippi, our old woman, she's further away from everybody else. And therefore I didn't include her much in group observations because she just doesn't interact with the other lynx. She's hanging out down there. But I remember hearing about Pippi when I was working at Home Range in 2024. They knew about her and they were keeping an eye on her. She's a sweet old lady and she's had many kittens, so she's just resting down south. More group observations. Like I said, Honey and Oscar are moving together and they have overlapping home ranges. They're young. It's mating season. Hopefully they're mating, which would be wonderful because like I said, lynx are endangered in the state of Washington. So the more kittens we can have, the better. And there are periods of time where I saw that the lynx were moving and periods of time where I saw that they weren't. So these were just sort of questions that I had about that. Do they have a feeding schedule? Are they able to cache food for a couple weeks or a week at a time and then they need to go hunt again? Was there bad weather? I don't know what it looked like. Maybe they don't want to get wet if it's raining a lot and they want to just hang out in a den sleeping. They can be nocturnal, they can move around at night, but maybe they don't want to. Maybe they want to sleep. Um, and yes, so like I also said earlier, Freddie and Honey were captured in the same spot, but their GPS signals don't show them overlapping over the course of the month. So that's just to exemplify that their home ranges are vast and that this only captures a small percentage of it. And then I'll end on some sort of implications and like wider reaches of this project, and what I was able to find out. So this is zoomed in a little bit of Unit 4 for that critical habitat. Critical habitat is a designation that's required by the Endangered Species Act once a species is listed as threatened or endangered. So that's why we have this habitat here in Washington. And it kind of means that, oh, this is where lynx were found when they were listed as threatened by the federal government, or it includes habitat that is important or habitat types that are critical to lynx. Like I said, they like what they like and they don't really want to live outside of those areas. So if there are areas that include habitat that's important and critical to them, it's important for conservation efforts that we can kind of keep it that way. And that's what Carmen and Anna are doing over at Home Range with their Lynx and Wildfire study. They really want to see if lynx are using that tripod burn area, because if they are, then that's great. That means that their habitat can maintain as such. But if not, then that could be extremely detrimental to the population that's living here. And it could have huge implications moving forward, since I'm sure we all know large scale wildfires are becoming a huge problem due to climate change and fire suppression techniques over the years. So if lynx aren't able to use areas that have been recently burned, that would be a big problem because it means their available habitat is even smaller. And then this is the lynx den I found. It doesn't really look like anything. And I remember hiking like a couple miles and showing up and they were like, here's the den. And everyone was like, what? But it's just under here and we were able to find it by finding little bits of lynx hair. Yeah, the little kittens would just be raised under there with mom not going far, but getting food and coming back. And you can see that this is an area that was burned by the tripod complex fire. This is what's called a matchstick forest, and it's what most of the environment looks like out there. But these are new little lodgepole pines coming up. And it's good because it's good ground cover for the baby lynx as well as the snowshoe hares. I saw a lot of bunnies running around when I was out there. So we know that the food is there and that they like living in that area. And just in terms of how this connects to the work I was doing for this capstone, I mean, really it was just to practice using telemetry data and GIS. I'm not a GIS major or minor, anything like that, but I enjoy doing it in my classes, and I think it's a good way to translate raw like data that doesn't make a lot of sense into something that can be understood by lots of different people and be analyzed a little bit easier. Um, and also it's good just to see where these links are moving, where they're going. It's sweet to see if they're mating, that they are, because that's huge, you know. Like I said, these animals are endangered, so the more kittens, the merrier. And that's also what this Linston kind of signifies. Yeah, that's it. Thank you for coming. And once again, huge thank you to Home Range, Carmen, Anna, Clara, and all the interns I worked with, because this project wouldn't have been a project without them. So, yeah. Ask me or the links questions. Yes.